So I'm just continuing with the previous class, which was on applications of the techniques you have learned in RDT course in the field of medicine. And what we had discussed last time was that how it is really important. These techniques have been used in the field of disease diagnosis as well as therapy. So I told you that by PCR or the variations of PCR or reverse transcription PCR, or you can design probes, immunoprobes for localizing a gene or alteration in the chromosomes by FISH technique. I mean, there are many ways by which you can diagnose a particular disease. Even it's not obviously limited to DNA and RNA. You can also do immunoprobing. And then is I told you about DNA fingerprinting, and in gene therapy, I told you that how mainly I had focused on the story of insulin, that which different strategies and which different strains and organisms are being currently used for insulin production for therapeutic purposes. So today I start with the recombinant vaccines and a very brief idea about the other points that are mentioned here, which is molecular farming and suicide gene therapy, etc. So let me go to that slide. So this is molecular farming, which is simply that you use either farm animals. So in which case you say that it's animal farming. And when I say farming, it is PHARM that I'm talking about, not. So farming means production of pharmaceutical proteins, uh, or let's say proteins for pharmaceutical applications in farm animals. So then it's called animal farming. Otherwise, if it is made in plants, then it is called as plant farming. So the idea was that can we overexpress the recombinant proteins in the animals and harvest means the food products of which we consume. For example, mainly I would come back to it's the milk that is harvested, and then you can use it for therapeutic purposes, because obviously if you express your protein in these multicellular organisms, and you can imagine that from one sheep or goat how much milk is being harvested, so from that milk you can take out your recombinant protein and purify it. So it is just that in very large amount you can harvest your protein. So that was the idea behind animal farming. And yeah, so yeah, mainly it is that you can harvest it in large quantities. Only issue is that making these transgenic animals is it takes a lot of time and lot of experiments, and only few experiments they are successful. So making these animals requires a lot of money and patience and time. But once these are made, and then their progeny would have that gene, and then you can harvest the protein of your interest from any part that I'm telling. It could be blood, it could be the milk, etc. So these are the two examples which are given. So in this case, Tracy was the first mammal which was engineered to overexpress a protein called antitrypsin for the treatment of cystic fibrosis, and it was produced in the milk. And similarly, this is in 2009, the US FDA, they granted marketing approval for the first drug to be produced in genetically modified livestock, and in this case it was atrin, which is antithrombin protein, which was purified from the milk of modified goats, and marketing permission was also given. So it means that yes, there is a huge scope in this particular field, and many people, many scientists, they are trying to use various organisms, various multicellular organisms, and in this case the farm animals, for production of these proteins. So basically what is to be done is that you clone your gene downstream of a particular promoter. What that promoter would be would decide that in which tissue that particular gene will be expressed, because you introduce this gene in the oocyte. So the animal that is developed from this oocyte, so in all the cells that particular gene will be present, but where that gene will be expressed will be solely guided by the fact that it is downstream of which promoter. So in this case they have introduced it downstream of lactoglobulin promoter, so it means that it will be expressed in the milk of this sheep, and the milk can be harvested, and from that milk you can harvest your final protein. So apart from, okay, I don't have the slide now. So anyway, I just want to know that rather than this, okay, one slide is anyway missing. So it was that apart from milk, you can also harvest it from the blood. But the more common one is this milk only, and the common promoters that are generally used, they are beta lactoglobulin, or they are whey acidic protein, both of which give very high expression in the milk tissue, in the tissue which gives rise to milk. Then coming to plant farming, and here again the idea is that we can express, because after all these also are eukaryotic in nature, so we can express certain proteins in plants, and then harvest any part of the plant where it is expressed, or if you are expressing it into the whole plant, then you can harvest the whole plant. So here the point is that plants and animals, they have similar protein processing activities. Obviously, for depending on which protein you are working on, you have to figure out that the protein which is expressed in plant, is it exactly similar to the one which is expressed in animals? Because in some cases, because of some differences in the glycosylation pattern which we see in plants, there may be some differences between the plant and the animal uh, protein. So only in those cases you have to be little worried about, but otherwise recombinant proteins have been harvested from plants, and many transgenic crops such as maize, tobacco, rice and sugarcane, they have been genetically engineered to express these recombinant proteins for therapeutic purposes. So in plant farming also, what you do is that you take the DNA molecule, you introduce into plant cell. That introduction, if you remember, it could be either through agrobacterium tumefaciens, or it could be through the other techniques that I told you. It could be electroporation, it could be particle bombardment, or plant viruses. Many different ways by which you can introduce your DNA into the plant cell. Once it gets incorporated, and depending on which promoter you have used, you can harvest that particular plant part and isolate the desired protein, which will be finally used for pharmaceutical purposes. Now, coming to, can we only use means the therapeutic proteins? Does the scope of using these recombinant proteins is limited to only those proteins, or can we also make vaccines out of it? So definitely, see, the current vaccines which are in the market against various diseases, they are largely either they are weak strains of a particular virus, or what they have done is that they harvest the protein. I mean, they, depending on that, the which protein of the virus is involved in imparting or let's say activating the immune response within the human cell. So first that protein is identified, and once we identify that, okay, probably this coat protein or this surface protein is important for eliciting an immune response in the human body. So what we can also do is that we can make that protein and introduce into the humans with the idea that once this protein is introduced, it will elicit the immune response, and that can be used as a vaccine. For example, for coronavirus, you know that mRNA was used. So similarly, in many cases, proteins are also used for as vaccines against particular disease. So how these vaccines, which organisms are generally used for developing these recombinant vaccines, there are examples in which they have been synthesized in yeast, and then this protein was harvested and then it was given to the humans, or it was made in CHO, that is these mammalian cell lines that I told you. They are very good for recombinant protein production. They have been made in these and then given. The most common example is this hepatitis B surface antigen. So which protein or which combination of protein should be used for as vaccines is what you have to think about. For individual virus, you have to do a lot of studies and see that maybe in some cases it is not single protein but a combination of protein which is eliciting a better immune response. So all that thinking you have to do, but once you know that okay, this single protein or two proteins are important, then what you have to do is you have to overexpress it in these cells and then just purify it and provide it, means inject it into the human body. Then can means only the yeast or CHO cells have been used for making recombinant vaccines? No. The idea was also means this idea is also coming up that can we use plants as source of recombinant vaccines. The idea is simply that probably I'm interested in some means somebody has to be administered a vaccine. So how about that he just eats some plant and gets that recombinant protein in his body, which will be as a vaccine to it. So this is the idea about these immunity could be acquired simply by eating part or all of the transgenic plant, means it can be gajar, it can be muli. I mean anything that is eaten raw, that can be used as a source for recombinant vaccine. So, for how these vaccines are produced, exactly same strategy that I told you, that how these recombinant proteins are produced. You simply have to introduce a gene fragment into the plant cells, and that will make that particular protein. And only thing is that if let's say you want to make a edible vaccine from this any plant, I mean this radish or any these things, then you have to ensure that it is you use a promoter which is expressed in that particular plant part. If you are eating a leaf, then it is important that you are aware that that recombinant protein should be expressed under the control of a promoter which is very highly expressed in the leaf cells. Okay, so just want to say, if you are means eating the root of a particular plant, then you have to use a root specific promoter. These things you have to keep in mind. Now, what are the issues which are there for edible vaccines is one, that there could be variability in the yields between different plants in a single crop. Another point is that have we been able to produce sufficient amount of recombinant vaccine in these plants? So as you can read all these notes later, you can see that to be completely effective, the yield of the vaccine needs to be make up 8 to 10% of the soluble protein content of the part of the plant which is eaten. But in practice, yields were found to be much less, usually not more than 0.5%. So all these things have to be kept into mind, and a lot of changes these scientists are making to ensure that the protein that they want in higher quantities, how do they get more amount of it? So one idea was that if you introduce your gene of interest into the nuclear genome, then you get a single copy of that particular genome, that particular gene, right? Because nuclear genome is present in one copy. But if I introduce it into the chloroplast genome, all the plants, they have chloroplast, then because chloroplasts are present in multiple copies, so we have huge amount of protein that can be made if we overexpress our gene of interest into the chloroplast. So this was one idea. However, it may be not suitable for those gene of interest which are heavily glycosylated, because in chloroplast the proteins which are made, they are not glycosylated. So these are certain issues with respect to edible vaccines. Some aspect of it I'll revisit when I'll be telling you tomorrow of the RDT, the use of RDT technology in the field of agriculture. But yes, there are certain issues related to it, and scientists are trying to overcome these. And there are some experiments which have clearly shown that, for example, this example is given, Vibrio cholerae B subunit, which can be used to confer immunity against disease such as cholera. This protein was synthesized in transgenic tobacco, tomato, and rice plants. I would like to say that I would like to revisit that point, that overexpression of these proteins in dicot plants is very easy, because making transgenic plants in dicots is anyway easy, because through agrobacterial tumefacients you can make transgenic plants. But in monocots it is comparatively difficult. So many of the first experiments just which are as a proof of concept to show that indeed recombinant vaccines are effective. So in those cases they are generally done in dicot plants. You will always find that. So it was seen that the protein which was made in this, so it was shown to elicit anti-cholera immune response when leaves or fruits or seeds, they were fed to mice. Okay, so there is definitely scope of this, and then it will be really easy. Just by eating a particular plant part, you can get the vaccine. Then there is another way by which the recombinant vaccines can be given with the use of vaccinia virus. I'll just come to this point. But first, let me tell you that when these recombinant proteins they are made, let's say you know that surface coat protein of a particular virus that is able to elicit an immune response. So what you do is that you overexpress that particular virus protein in any of the organism that I told you. Let's say you overexpressed it in CHO cells or yeast cells or bacteria, whatever, and then you harvest that protein, and then what you do is you inject it into the bloodstream with the idea that the antibodies will be made against that particular protein by the human body, because it recognizes it as foreign. And when so once these antibodies are made, so then when later in life you somebody is exposed to that particular virus, then these antibodies are quickly made and they bind to this virus and then inactivate it, or destroy this virus. So this is the idea behind any vaccine, recombinant vaccine that is used. But it was seen that when the protein was introduced into the humans, just by, as a purified protein, that then the immune response was not very strong that it should have been when the whole virus was going or something like that. So because you want a very active or strong immune response, so that the antibodies are made against any future infection. But if the immune response is not strong enough, then you won't have all set of antibodies which are made against that particular protein, and we may get a comparatively weak immune response, which may be comparatively less effective in controlling the virus infection in future. So then it was realized that how about we clone this recombinant protein, we introduce the gene for this recombinant protein into a virus which gives very weak symptoms in the human. So in that context came this vaccinia virus, which when infects humans gives very, very weak symptoms, very mild symptoms. Okay. So it was realized that so what we do is that, let's say in this case it's a coat protein, you clone it within the vaccinia virus genome, and then you introduce these recombinant vaccinia virus into the human body. So then it was seen that when it was introduced as part of a whole virus particle, this protein, then the immune system, it was reacting much better as compared to this protein alone. So it was realized that okay, this strategy also can be very useful in making these recombinant vaccines. So obviously when this virus will infect human cells, then what will happen, the whole virus particle, including the protein that is being made by this viral genome, the foreign this surface protein, so all these will be made and recognized as foreign by the human body, and we will have anti-hepatitis B, in this case for example, antibody, as well as anti-vaccinia antibody. So then means this is a good way by which you can make recombinant vaccine using vaccinia virus. So some of the foreign genes that have been expressed in recombinant vaccinia virus, you can see that it's a good list that is shown here. And obviously over time, the list of the proteins that can be expressed in recombinant vaccinia virus and used as recombinant vaccine, it will increase. Okay, now, so what I've told you till now is the fact that yes, you can use recombinant proteins. Uh, they can be therapeutic or they can be used as vaccines in various sources, and that can be used in the field of medicine for treatment or for vaccine production, etc. Then coming to gene therapy, another important aspect of RDT, which is that you, to correct a particular disease, you introduce certain genes. So if you have a gene, you know that a particular disease happens because of the deficiency of a particular protein, then what you do is that you introduce that particular gene in the organism, so that will be overexpressed, and then we assume that the disease will be corrected. So it is generally used and found to be very useful, especially for those diseases which are single, let's say monogenic diseases. However, for multigenic diseases, you have to figure out that among the multiple genes, for example, in cancer, which are aberrantly, you know, which are altered in cancer, which are the genes, the overexpression of which will be really useful for treatment of cancer. You have to do some pre-studies to ensure that out of the various 1,000 genes which are altered in cancer, which genes will be the key genes, overexpression or inhibition of which will cure that particular disease. Okay. So in gene therapy, we have this overexpression approaches, and now even this inhibition approach is also counted as gene therapy. That in those diseases where we know that overexpression of some abnormal protein, or the even overexpression of a wild type protein is leading to a particular disease, then by inhibition of that particular gene by siRNA or microRNA approach, can we treat that disease or not. So now when we talk about gene therapy, there are two particular therapies that we are, terms that we are aware of. One is germline therapy and one is somatic cell therapy. So currently for treatment of various diseases, it is only the somatic cell therapy that is being used, and germline therapy is generally not used because there is a lot of ethical issues associated with it. So because in germline, see in somatic cell therapy, what you do is that you take somatic cells, you correct a particular gene. If you want to overexpress it, you overexpress it, or you want to inhibit it, you inhibit a particular gene, but it is in a somatic cell. So obviously if it is in a somatic cell, it will not be passed on to the progeny. So there is no ethics related to it. But in germline therapy, what is done is a fertilized egg is provided with a copy of the correct version of the gene of interest and reimplanted into the mother. If successful, the gene is present and expressed in all cells of the resulting individual. So if you know that probably the to be born child, he will have mutation in a particular gene and you want to correct it. So one way is that you take the egg, you correct that particular gene, you reimplant it into the mother, with the assumption that the child that will be born will no more have that particular disease, that maybe by CRISPR you have corrected that mutation, or you have overexpressed a gene, or you have deleted a abnormal gene, whichever way. But then you must be aware that there are many ethical issues which are related to it. Many of these, they are pointed out here, so you can please go through those. So the main thing is that because whatever changes you are doing in germline therapy, they will be passed on to the progeny and so on. So there are ethical issues. It could be that how can you be sure that whatever changes you are making in that particular organism, whether they are good changes or bad changes, who decides that? Maybe you think that you are doing a very good job by altering a gene, but you know that there are many gene interactions in the body. Maybe you think that you will be correcting one particular disease, but then maybe by you might not be aware that alteration of that particular gene may also lead to some other changes in the body which are undesired and so on. And should people be allowed to use gene therapy to enhance basic human traits like beauty, intelligence, athletic ability? Will finally the use of gene therapy make society less accepting of people who are different? Will gene therapy, because of the high cost associated with it, will only be available to the wealthy, and the poor will not be able to avail this facility? Who decides which traits are normal and which constitute a disability or disorder, etc. So you must be aware. I had told you about that CRISPR story where a Chinese scientist, he was arrested finally because he made changes in the to be born child by CRISPR technology, and with the assumption that the babies will not be suffering from AIDS in future, but then it was ethically wrong, etc. Anyway, so other one is the somatic cell therapy, which is generally used, and there are two ways by which you can do that. So let's say a particular uh, there is some patient suffering from a particular disease, and you know that changes in the expression of a particular gene will lead to treatment of that particular disease. So what you do is that in the bloodstream you inject that particular, it could be a vector or it could be a protein, and we assume that that particular gene will go to the target cells, and then the overexpression or inhibition of that particular protein will take place. So that is comparatively more used therapy as compared to the gene therapy. And another option is, and somatic cell therapy, it is found to be very valuable for especially the blood diseases, because in this blood diseases, the option is that you take the blood stem cells, you isolate, because you can isolate the blood, you can harvest the cells which are defective, you can transfect them with the new gene, or you can silence certain genes in the isolated hematopoietic stem cell, that is blood stem cell. You correct the whatever aberrations there are, and then you just reimplant it into the human body, the patient's body, with the assumption that now, because the corrected cells are present, and these are the cells which give rise to all the blood cells in the body. So the person who is suffering from a particular blood disease, it could be hemophilia or thalassemia, he will be corrected for that particular ailment. So it shows most promise for definitely the inherited blood disease. And there is also some research going on that this therapy also be used for lung diseases, because it was realized that if we clone the DNA, we introduce a recombinant DNA through adenovirus, then it is very well taken up by the epithelial cells in the lung by liposomes. So it means that if the lung cells, they can easily take up the corrected gene, then the ailment, it can be corrected. But it was found that it requires repeated treatment with the same, because the gene expression occurs only for a few weeks. But obviously a lot of research is going on in this field, and in future it could be that we get a permanent treatment, not only through the blood cells, but also through other cells which can get corrected by gene therapy. Only thing is that it has to be highly targeted in nature. In case of blood cells, we don't have to worry much, because you just take out the target cells, in this case the hematopoietic stem cells, and correct it. But when we are giving these siRNAs, microRNAs, or these overexpression constructs into the body to correct a particular disease, you have to ensure that it only goes to the target cell, but not to the normal cells. Means there should be minimal side effects in the normal cells, and it should be highly targeted in nature. That is very important point that has to be kept in mind. I told you that gene inhibition approach, you have also been taught this in the gene assignments. So there are two methods to do this. So if you know that a particular gene is highly overexpressed and you want to you want to normalize its level, then one way is that you design a siRNA against that particular gene, with the assumption that that siRNA will go and bind to its target transcript, and then it will bring about the mRNA cleavage, because the siRNA, it will recruit the RNA induced silencing complex, which will make cuts in the mRNA against which it was designed, and that will lead to cleavage of the mRNA and normalization of the levels. And similarly for microRNA also, we know that microRNAs, when they are introduced into the cell, they go and bind to the target gene. Only thing is that in case of microRNA, it is not related to one gene targeted, but it targets multiple genes. So in many cases, multigenic disorders where you want to alter multiple genes at the same time, you may go for microRNA based approach, where you overexpress a microRNA which will go into the cell. It will go to various different target mRNAs where it binds, and then it also recruits the RNA induced silencing complex, which includes these proteins, dicer, etc. And it includes various RNase proteins, and brings about the degradation of the target transcripts. So this is also part of gene therapy. So both gene overexpression or inhibition approaches. Then lastly, coming to the suicide gene therapy. So what we have understood till now, that okay, if there is a cell where the gene is deficient, you can overexpress it. If it is overexpressed, you can inhibit it by siRNA or microRNA approach. And there is also option that you can target, you can induce the death of the diseased cell, right? So here the idea is, can we introduce a toxic protein into the target cell, let's say it's a cancer cell, and thus lead it to commit suicide, or die, induce death in the disease cells. So here what is done is that you identify a particular toxic protein, which you know for sure that when it is introduced into a cell, it will definitely lead to the death of the cell. Most of these toxic proteins, they are the ones which affect the translation of the whole translation process, etc. So definitely the cell will die. So only thing is that when you go for such type of therapy, you have to be 100% sure that it goes to the target cell only, because you do not want that it should bind even slightly to the normal cells. So in this case, when you want to go for suicide gene therapy, the monoclonal antibodies, that is the antibodies which are extremely specific to the target cells. Let's say you have to know that which are the proteins, which are the surface proteins which are present only in the cancer cells or any other disease cells, and that antibody is hooked on to the toxic proteins. So the idea is that when you introduce those antibodies in the body, so those antibodies will go, let's say, only to the cancer cells and deliver, they will get internalized and deliver the toxic protein into the cancer cell, which will lead to its death. So this is called suicide gene therapy. Alternatively, another option is that you prime the body in such a way that it targets the cancer cells or disease cells very, very efficiently. That is, can be train the patient's immune system in such a way that it targets, let's say in this case, tumor cell, or maybe in other diseases, some other disease cell, by priming the human body in such a way that it synthesizes either the tumor cell, it synthesizes strong antigens, or we train the B cells or T cells in such a way that now they are able to recognize the tumor cells very, very efficiently, because we know that in general the tumor cells, they try to evade the immune system by various strategies, but scientists have found a way by which they evade, and they are what they are doing is that they take these blood cells out, including the white blood cells out, I mean, and then they make changes in that, and they in a way train them that if this protein is now produced in those T cells, now they can very efficiently recognize these tumor cells and bring about their death. So this is another way by which you can target the disease cells. And again, this involves cloning. So that's why it's part of this lecture. So in this case, what they do is they take out the T cells and introduce certain genes which can help in killing these tumor cells much better. So that's all for today's class, and I'm open for questions. Okay, so if there are no more questions, okay, then have a good day and bye bye.
Thank you,
Thank
ma'am.
you.
And we'll meet
Thank
tomorrow.
you, ma'am.
Okay, we'll meet tomorrow, same time at 9 a.m.